Hi, welcome. My name is Sameek Roychowdhury. I'm an associate professor of medical oncology and a cholangiocarcinoma researcher at Ohio State University in Columbus, Ohio. And it's my pleasure today to be a moderator for our International Cholangiocarcinoma Research Network monthly seminar on topics related to cholangiocarcinoma. And so I'm pleased to introduce, and I'll let him tell you a little bit about himself, Javier. Javier?
Hello, thank you, Sameek, for introducing me. Thank you also to the ICRN and the ENS-CCA for giving me the opportunity to present our work here in this interesting seminar series. I am Javier Vaquero. I am a researcher at the Bellvitge Biomedical Research Institute here at Barcelona. But today I'm going to show you a work that I started when I was a postdoc at Saint Antoine Research Center in Paris under the supervision of Laura Fossier. And it's a project that we published last year, but that we are continuing now, working more deep in this subject about ZEB1 here at Barcelona in collaboration with my former group in Paris. So today I'm going to talk to you about the role of ZEB1 in regulation of the tumor-stroma crosstalk in cholangiocarcinoma. So we are going to talk about cholangiocarcinoma, but since we are a group that works on this, I'm not going to go a lot deeper into this. I'm going to talk about the stroma, because one of the main features that are important for this talk regarding cholangiocarcinoma is desmoplastic characteristic. It has a very prominent stroma formed by multiple type of cells. And during the last years, many groups have been involved in projects trying to understand the role of these different populations from the immune compartment, the endothelial compartment, in the progression of cholangiocarcinoma. But due to its central role, the CAFs have been the compartment or the cell type more deeply studied during the last years. We have been involved in this research, but lately, in the last couple of years, with the arrival of single-cell transcriptomics, this subject has been even more complicated, because these two groups have performed single-cell transcriptomics to describe that the CAF population is not a unique population, but is even more complex, and there are different subtypes of CAFs with different signatures that allows them to do different functions. So this is getting more complicated every year. However, the protagonist today is ZEB1. ZEB1 is a single-thing EVOX binding transcription factor. It can act as a transcriptional repressor of usually epithelial genes, or a transcriptional activator, usually of mesenchymal genes, depending on the complexes that format the promoter of the target genes. And it regulates epithelial and mesenchymal genes because it is an EMT-inducing transcription factor. So the EMT, or epithelial to mesenchymal transition, is a dynamic and reversible process in which the cells lose the epithelial characteristics and gain mesenchymal proteins to acquire migratory and invasive properties that allows them to get more metastatic. So this process is regulated by different families of EMT-inducing transcription factors. So you may be wondering why we choose ZEB1 and not one or the others. This is because many reasons. First of all, in a previous study that we performed in human cholangiocarcinoma, we studied the resistance of cholangiocarcinoma cells to anti-EGFR therapy, and we observed that our resistant cells activated the EMT process to become more resistant to the drugs. And one of the more prominent transcription factors that regulated this process in our resistant cells was ZEB1, as you can see here. Also, we were interested in ZEB1 because it not only regulates EMT, but many other different processes, such as cancer stemness, such as DNA repair, or even immune escape. But also at the same time, we were characterizing our cellular models at the lab regarding the expression of different epithelial mesenchymal markers, and we observed that in our panel of cells, we have very nice model with very epithelial cells that do not express ZEB1, very mesenchymal cells that express high amounts of ZEB1, and that this expression correlated very well with the EMT markers. But also at the same time, we were writing a review on this topic, on epithelial to mesenchymal transition in cholangiocarcinoma, and we observed that when we were writing this, there was pretty much no mention about ZEB1 in the literature regarding its role in cholangiocarcinoma. So for us, the question was, why not ZEB1? So we started this project with the aim to unravel the role of ZEB1 in cholangiocarcinoma, and we started working with tumor cells. But along the way, we discovered that fibroblasts were involved in this process and that ZEB1 was a regulator of this tumor-stroma crosstalk, as you are going to see in the next slides. So we started this analysis by performing, in collaboration with Valérie Paradis and Nathalie Getsch from Bouillon Hospital, an immunohistochemistry study analyzing the expression of ZEB1 in a panel of 45 human cholangiocarcinomas. What we observed was that it was not expressed in normal cholangiocytes from adjacent non-tumoral liver, but it was expressed in human cholangiocarcinoma, in tumor cells from cholangiocarcinoma, and its expression increased in poorly differentiated cholangiocarcinoma. And its expression correlated also with tumor grade and with vascular invasion. This correlated with our in vitro observations because the more epithelial cells, if you go to the original publications, were derived from more differentiated tumors, whereas the more mesenchymal cell lines with high expression of ZEB1 were originated from less differentiated tumors. And this correlated also with transcriptomic analysis that we performed from the public database from the TGF-Beta cohort, and also from what we call the REN cohort that we did in collaboration with Cédric Coulard and Corentin Louis at Université de Rennes. So the expression of ZEB1 correlated negatively in human patients with the expression of epithelial markers such as CDH1, that is the gene encoding for e-coloring, or ESRP1, and correlated very positively with the expression of the typical mesenchymal marker BMP. So
Javier,
to go more deeply, yep.
Javier, thank you. This is great. If I can ask you a question,
Yes, of course.
Be kind of interactive. So in this first two figures in your paper and what you're talking about today, you've looked at human cholangiocarcinoma and C expression. If you go back just one slide here, there's a variety, right? There's some that are a little bit higher than others, and we know from the studies here, these cohorts, that we've got a variety of cholangiocarcinoma subtypes. So do you see a different pattern of expression for intrahepatic or extrahepatic cholangiocarcinoma, or perhaps gallbladder is probably included in these cohorts too? So do you see is one preferentially the top expressing for ZEB1?
Yeah, actually, we don't know about that. It's something really interesting that we would like to know better. But actually, these cohorts for the TGF-Beta cohort, I think is 39 patients or something like that. And most of the patients, I don't know if it's 31, as intrahepatic cholangiocarcinoma. And for the REN cohort, all of these patients have intrahepatic cholangiocarcinoma. So right now, our human data is more concentrated on intrahepatic cholangiocarcinoma.
Yeah.
However, in our
and then you've got a band of expression, right? There's some that are high and some that are medium and some that are low. So it'd be interesting to see. Anyway, so thank you.
Yeah. Anyway, if you see at the cell lines, we have intrahepatic and extrahepatic cell lines that express ZEB1. So at least it can be expressed. I don't know at what extent, but at least it may be there. So as I was saying, we decided to go more deeper in the understanding of what ZEB1 was doing in tumor cells, and we generated different cellular models by genetically modifying our cell lines. So we took HCCT1, that is a cell line that do not express ZEB1, and we overexpress it by infections. Then we took a cell line that highly expressed ZEB1, and we downregulated its expression, but also by lentiviral infection. And then we took advantage of our previous publication in which we generated, as I told you, resistant cell lines. And these resistant cell lines naturally overexpress ZEB1 as a cause of the resistance. So we took this resistant cell line and we downregulated ZEB1 again to have like kind of a mirror experiment or mirror model with the overexpressing one. For the rest of the presentation, to simplify the results, I'm going to show you the results with this model mainly, but we got similar results for the other three type of cellular models. So first of all, we wanted to analyze if indeed modulating the expression of ZEB1 modulates the EMT in these cells. And indeed, the upregulation of ZEB1 led to an downregulation of epithelial markers, CDH1 and ESRB1, and an upregulation of the mesenchymal marker BMT. But as I told you, ZEB1 does not only regulate EMT, it regulates many other things, and we were interested in the stem phenotype of the cells. And it has been described that a particular population of cells that share a signature of two typical stem markers that are CD44 and CD24, a signature of CD44 high and CD24 low, mark a population of cancer stem cells that share also EMT traits. So we analyzed this in our cells, and we observed that, again, the upregulation of ZEB1 increased the expression of CD44 and downregulated strongly CD24. And this population of with this specific signature was increased in our ZEB1 overexpressing cells. However, at this point, we only had expression analysis, so we wanted to see if these expression changes were translated functionally to our cells. And actually, when we perform functional studies, we observed that the ZEB1 overexpressing cells increase the migration, increase the color information, and increase the ability to generate more and bigger spheres. So somehow our cells were becoming more aggressive. But to validate this, we wanted to evaluate how our cells behave in an in vivo environment. And to do this, we came to the xenograph model, and we injected under the skin of the immunodeficient mice both control or ZEB1 overexpressing HCT1 cells. The thing about this model is that, according to our pathologist, the histology of these xenographs resembles a human cholangiocarcinoma. But this is more interesting because the architecture of this model means that the tumor cells are from human origin because we inject cell lines of human origin, but the murine cells that are recruited to form the stroma are from murine origin. So if you design human primers or murine primers, you can analyze gene expression in the tumor cells or in the stroma. So for the rest of the presentation, to indicate what we are doing or what we are showing, I will put these symbols when we are showing gene expression in these xenographs, and I will put these symbols when we are showing murine gene expression from the stroma. So actually, we observed when we performed the experiment that our ZEB1 overexpressing tumors generated bigger tumors. And at the histological level, it seemed that these tumors were less differentiated and had a more prominent stroma. Red series staining confirmed the more or increased the position of extracellular matrix. And actually, when we evaluated the expression of murine markers for typical fibroelastic markers, we observed an increase in the alpha-SMAM gene and in the expression of collagen 1 and collagen 4 genes. So the next step was to try to understand how ZEB1 overexpressing cells what were they doing to generate bigger tumors and to increase the proliferation of stromal cells. And for this, we analyzed, in this case, the expression of genes in the tumor compartment of the xenografts, and we evaluated the expression of the main profibrogenic factors that are TGF-Beta 1, CCN2, that is the gene encoding for CTGF, and PDGF-B. And what we observed is that only the expression of CTGF was increased both by mRNA and by Western blot, as you can see here at the protein levels. So the expression of CTGF in the tumor cell may explain the increased stroma. But to validate this, we wanted first to validate that ZEB1 may be regulating somehow the expression of CTGF. And actually, when you see the expression of CTGF, it correlates very nicely with the expression of ZEB1 in our panel of cells. It also correlates with the expression of ZEB1 in our genetically modified cellular models. When you overexpress ZEB1, you overexpress CCN2, and the other way around when you don't regulate ZEB1. And also the expression of ZEB1 correlated with the expression of CCN2 in our two cohorts, the RAIN cohort and the TCGA. But furthermore, it had been described that ZEB1 together with JAP may become the activator of certain genes by binding to a specific sites different to the general e-box that ZEB1 binds normally. And we wanted to validate this by chromatin immunoprecipitation assays, because indeed JAP signaling is prominent in cholangiocarcinoma. So when we performed these experiments, we indeed observed that ZEB1 was able to bind and to upregulate the expression of CCN2 promoter. Next,
Hey,
to give some
Javier,
kind of, yep.
if I could ask a question on the previous slide. So far, we've seen that the ZEB1 expression in this low expressing cell line changes some of its features for migration and colony forming.
Yep.
And now you've shown this in xenografts in the same cell line. And now you've demonstrated a target gene of ZEB1, which is CCN2. Do you think there could be other target genes that are important besides CCN2?
Yes.
A transcription factor. Any thoughts on what else could be important for ZEB1? And then at the beginning, when you explained, why you chose ZEB1, there was some evidence that this was involved in other settings for EMT. I wonder what we could learn from other cancer types for ZEB1 and EMT. So what do we know about EMT for ZEB1 and other cancer types, and also, what other target genes could be affected by ZEB1 here?
Yeah, indeed. Yes, sure. ZEB1 is doing more things. You will see during the presentation that we discover some other target genes of ZEB1 that are also growth factors. But the thing is that, if I am honest, we never had a specific funding for this project, so we could never perform any big transcriptomic analysis to evaluate all the target genes that ZEB1 may be regulating in these cell types. But the thing is that ZEB1 is a transcription factor, so it regulates a plethora of function in the cells. This is one of the reasons why we choose ZEB1 to perform this study. Actually, I have been asked many times why I choose ZEB1. This is why I made a specific slide in the introduction, because in the lab, since ZEB1 is not a targetable gene right now, I got all the time the question, why ZEB1, why ZEB1, why not something else that you can target with drugs? And actually, the study of ZEB1 may give rise to actionable targets, as you can see here with CCN2 and further with other genes. And regarding other tumors, the thing is that ZEB1 does kind of everything in the tumor cell and in other cells from the stroma. ZEB1 regulates EMT, regulates metastasis, regulates immunosuppression. So if we go deeper into the understanding of the ZEB1 network, we will find many more actionable targets than CCN2 or the other ones that I'm going to show you today. And that is why we want to keep working on these transcription factors, because we think that is something interesting and that can help, as you will see in the last part. I think we can help to improve the treatment of patients targeting ZEB1 related molecular mechanisms. So to give some kind of functional importance to CTGF, we perform proliferation or cell viability studies with fibroblasts. And what we observed actually is that CTGF was inducing the proliferation of fibroblasts, but also when we perform condition media to validate that tumor cells were producing CTGF, we observed that actually condition media from ZEB1 overexpressing cells induce the proliferation of our fibroblastic cells. But also when we included in the condition media a neutralizing antibody against CTGF, we saw a significant decrease in the viability of the fibroblast, although not complete reduction of the viability, which means, as you were actually asking before, that CTGF is here in the condition media, is produced in the extracellular media of these tumor cells, but there are probably some other factors that are involved in increasing the desmoplastic characteristic of this stroma in our xenograph models, at least. At this point, we knew that overexpressing ZEB1 in the tumor cells that may happen during tumor progression in a human patient make the cells become more aggressive. They develop EMT and they develop stemness. And these more aggressive cells secrete factors that help the tumor stroma to grow, and probably, as you will see after, the tumor stroma secrete other things to help back to the tumor cells and to increase tumor progression. But let's now go ahead. So to continue, we focus more on the fibroblasts, and we focus more on the fibroblasts because in our immunohistochemical study that I showed you at the beginning, we observed that on top of the ZEB1 expression that we saw in tumor cells, we saw also ZEB1 expression in the stroma. And we were very interested about this. And what we did was, again, in collaboration with Valeria and with Natalie Goetz, we performed co-immunohistochemical studies marking or labeling ZEB1 in brown and alpha-SMA in pink. So you can see here that there are cells in the stroma that co-express both alpha-SMA and ZEB1, meaning that ZEB1 is expressed by cancer-associated fibroblasts in cholangiocarcinoma. Also, we found expression of other cells in the stroma that do not express alpha-SMA, so it means that maybe other cells that express ZEB1 in the stroma. But focusing on the calves, we went back again to our collaborators from REN because they have a cohort of laser microdissected stroma from cholangiocarcinoma. And what we observed is that, again, the ZEB1 expression in this stroma from cholangiocarcinoma correlates very well with markers with profibrogenic markers such as TGF-Beta or CCN2, and with typical markers of activation such as alpha-SMA or extracellular deposition like collagen 41. So to go deeper into this, we went back in vitro and we generated a new cellular model. We took this cell line that I showed you before. It's called H-TERT and it's derived from hepatic stellate cells. So it's a liver fibroblast. And we downregulated ZEB1 because, as you can see here, and corroborating our findings in the immunohistochemical studies, these cells express high levels of ZEB1. So when we downregulated ZEB1, we observed that, again, the expression of the profibrogenic markers and the expression of the alpha-SMA and collagen 4 was strongly decreased in our cells. Again, we went back to the in vivo to see how this different expression of ZEB1 in the fibroblasts could impact tumor progression. In this case, we choose another cell line. We choose EGA1 to have a very epithelial cell line that do not express any of these EMT factors. So we don't have interference with our fibroblasts, and we co-inject them with either control fibroblasts expressing high ZEB1 or SH-ZEB1 downregulated fibroblasts. And what we observed is that indeed when EGA1 cells, when tumor cells were co-injected with ZEB1 downregulated fibroblasts, the tumors grow less, the tumor progression was reduced compared with the control tumors. And what was even more interesting is that when we analyze the expression of genes in the stroma, in the murine stroma of the mice, of the tumor, we observe again a downregulation of all these genes, a downregulation of ZEB1 and a downregulation of all the markers that we showed you before, TGF-Beta, CTGF, PGF-B, alpha-SMA, and collagen 4, which means that these fibroblasts with different expression of ZEB1 that we injected into the mice may be interacting either with tumor cells to educate fibroblasts or directly educating the murine cells to recruit them for the stroma. So to investigate a little bit more what was the fibroblast, or what were the fibroblasts doing to the tumor cells to increase the growth of the tumors, we performed condition media experiments. And actually we saw that when we expose tumor cells with condition media from control fibroblasts expressing high ZEB1, the viability or the proliferation of the cells increased significantly, and this effect was impaired partially or totally when we incubate them with condition media from fibroblasts depleted for ZEB1. And this correlated very nicely with the activation of intracellular signaling pathways such as STAT3 or AKT. So at this point, we knew that ZEB1, both in the tumor cells and in the stromal cells, was somehow regulating the crosstalk between these two types of cells. So we wanted to go deeper and to try to find some of these factors that you mentioned before that may be being produced either by tumor cells or by fibroblasts in response to the expression of ZEB1, and that could be involved in the progression of cholangiocarcinoma. For this, we organized our cellular, our in vitro and in vivo models. We took the overexpressing HCCT1 cells. We took the cells from our previous study, the HCCT1 wild type and resistant cells that, if you remember, overexpress ZEB1 naturally as a cause of the resistance. And also we had from our previous study xenograph experiments done with these cells. And we took also our downregulated fibroblasts and our co-injection study. We evaluated a panel of markers that I will tell you right after in our in vitro cells and also in the tumor compartment, tumor human compartment, and mouse stroma compartment in our different xenograph models. And what we analyzed was the expression of a panel, a wide panel of growth factors and cytokines that have been described to have an important role in tumor progression. And from this wide panel, we selected two genes that, or two proteins that gave interesting results, that are interleukin-6 and HGF, as you are going to see next. First of all, regarding interleukin-6, regarding the tumor cells, we didn't see any significant change because there was some tendencies in vitro to an upregulation in ZEB1 overexpressing cells. But when the cells were injected in vivo, this regulation disappeared, which means that there will be other mechanisms that regulate the expression of IL-6 more important than ZEB1. However, in our fibroblastic model, we observe a very, very strong regulation, downregulation of IL-6 in ZEB1 downregulated fibroblasts, and this happened again in vivo in the murine stroma. There was a downregulation of IL-6. So we wanted to validate if actually ZEB1 may be regulating IL-6 because it has been described in breast cancer cells that ZEB1 may regulate directly by binding to the promoter IL-6 and also IL-8. So for this, we took our fibroblasts, our control and downregulated fibroblasts for ZEB1, and we treated them with the main profibrogenic factor, that is TGF-Beta 1. Sorry, but the legend, I missed the legend, is not here, but this is TGF-Beta treatment. And what we observed is that TGF-Beta treatment induced a significant increase of both ZEB1 and in parallel IL-6 in control cells, but that this was completely impaired in both ZEB1 and IL-6 expression in SH ZEB1 downregulated fibroblasts, which means that indeed ZEB1 may be regulating IL-6 in liver myofibroblasts. But to see how could this be involved in the progression of cholangiocarcinoma or in the growth of tumor cells, we exposed to IL-6 to a panel of cell lines, both tumor cells and also fibroblastic cells. And interestingly, we only saw an increase in the viability or proliferation of tumor cells of this line of HCCT1 with IL-6, but not all the other cell lines. This is interesting because in one of the papers I showed you at the beginning, that they performed single-cell transcriptomics, they describe a population of calves that produce high amounts of IL-6, and they describe that this IL-6 is able to promote the proliferation and the stemness of cholangiocarcinoma cells. And this is interesting because the cell line used for these in vitro studies in this paper is also HCCT1 cells, which means that depending on the tumor cell that you are using, you may get some other effects because of the probably of the receptors that they express or the intracellular signaling pathways. And also this is interesting because us and also this group, they only investigated, and we only investigated the role of IL-6 in increasing the proliferation or the growth of tumor cells. But IL-6 is for sure having other functions and other roles in the tumor stroma because it's a pro-inflammatory cytokine. It has been described also in breast cancer cells that the upregulation by ZEB1 of IL-6 induces immune escape by attracting or by recruiting myeloid suppressor cells. So this expression of IL-6 may be having much more impact than we thought or that we think in cholangiocarcinoma.
Javier,
yep.
I know you're going to talk about another protein next, but for IL-6, that's really interesting. You can imagine that, a process of healing and inflammation and liver injury, the role of IL-6 in sort of normal liver function could be healing, right? And to drive hepatocytes or cholangiocytes in a recovery process. A simple question, do they express IL-6 receptor?
Yes, actually, is one of the first thing we did, because with all these experiments and the experiments I'm going to show after with the other protein, we wanted to know why some of the cells respond and why the other cells didn't respond. And actually, all the cell lines that we tested express IL-6 receptor and the partner of IL-6 that I don't remember right now, but it's like a pterodimer, and they express high amounts and similar to the HCCD1. So I don't know if it's because other interacting partners of the complex or some missing factors in the intracellular signaling pathway, but we don't really know. But it's not mediated by the expression of the receptors.
Got it. Interesting. Okay.
So then we went to the other factor, that is HGF, that was really interesting for us because it was involved in many things. So first of all, we observed that our ZEB1 overexpressing tumor cells express high levels of HGF, and that this expression was increased also when you put the cells into the mice, when the cells are in an in vivo environment. So this correlated also with the transcriptomic experiments or with the transcriptomic analysis, because ZEB1 correlated very nicely with HGF in both cohorts. And actually, to validate if ZEB1, because this was not described before, to validate that ZEB1 is a direct regulator of HGF, we perform again chromatin immunoprecipitation studies. And actually, ZEB1 binds to the promoter of HGF in tumor cells, and that is why it's able to regulate its expression. But also, we observe, surprisingly, that the stroma of these tumors also express more HGF when you inject tumor cells that overexpress ZEB1. But interestingly, in our in vitro model of fibroblasts, there seemed to be no regulation of HGF by ZEB1, because when you don't regulate ZEB1 in the fibroblasts, there is no regulation of HGF. So our hypothesis here was that tumor cells that overexpress ZEB1 may be secreting something that makes fibroblasts to produce more levels of HGF. So we validated this with condition media experiments, and actually we don't know yet which are the factors contained in this media, but actually when you expose fibroblasts to condition media from overexpressing tumor cells, HGF is highly induced. So again, we perform functional studies with viability studies to give some kind of important role of HGF here. And we observed two important things. First of all, in this case, again, answering to your previous question, all of these cells express HGF receptor. And we observed that in general, all these cell lines, or almost all these cell lines, respond increasing their proliferation to treatment with HGF. But what was more interesting for us is that the sensitivity of these cells to HGF seemed to be regulated by the expression of ZEB1 in the cells, because control HCCD1 cells that do not express ZEB1 do not respond to HGF, but the ZEB1 expressing cells do respond. And in the fibroblasts is the other way. The cells that express, the control cells that express ZEB1 respond, but the cells downregulated for ZEB1 do not respond. We have validated this with condition media. Condition media from fibroblasts is able to induce the proliferation of tumor cells, and tumor cells that overexpress ZEB1 respond better to this condition media. And again, when you take condition media from tumor cells and you treat fibroblasts, control fibroblasts with ZEB1 are able to respond, increasing the proliferation, but not ZEB1 downregulated fibroblasts. Now we don't know. Sorry, I always forgot about this slide. Also, we observed that HGF, treatment with HGF, is able to mildly activate the fibroblasts, because this is able to increase the expression of TGF-Beta, PDGF-Beta, and also alpha-SMA, and there is a tendency in CTGF and collagen 41. And again, this mild activation is dependent on the expression in the fibroblasts of ZEB1. So, as I was saying, we are not sure yet because we have not validated this, but we have some kind of subject or some kind of protein that may be related with this differential response, and this is integrin beta 3, because Integrin beta-3 has been described to be able to heterodimerize with many many growth factor receptor at the plasma membrane, and among them, interestingly, it can interact with CTGF, and it can interact also with MET, that is the receptor for HGF. And what we have observed is that ZEB1 overexpressing cells highly overexpress integrin beta-3, which may explain this response, and ZEB1 non-regulated fibroblasts express less integrin beta-3, which may be also involved in this differential regulation. But we still have to further validate this by siRNA experiments or other kind of experiments. This is interesting, again, because in the other paper I showed you at the beginning, is a nice study by Sylvia Affo and Roychowdhury group. They perform single-cell transcriptomics in human cholangiocarcinoma in different models, or in different mouse models, and they describe another population of calves that produce high amounts of HGF, and they say that this HGF is able to increase the proliferation of and the growth of tumor cells. And here we demonstrate also that this is not the only way, that the other way around can happen, and then tumor cells can produce also HGF to increase the proliferation and the activation of the tumor-associated fibroblasts. So at this point, answering to your previous question, we knew that, first of all, ZEB1 was involved in the activation of CAFs, and that some of these factors that were involved in this tumor crosstalk that were regulated by ZEB1 were HGF and IL-6. I'm sure that there are many more things here going on, but at least we have described these three interacting partners. And all of this crosstalk is for sure involved in the progression of cholangiocarcinoma. And also this is interesting because these are actionable targets. For example, CTGF, there is an antibody, neutralizing antibody against CTGF that has reached clinical trials in pancreatic cancer, for example. So maybe this could be an actionable target in the future of cholangiocarcinoma.
So for this diagram, I think you've nicely summarized the potential kind of secreted factors that are at play in this tumor and fibroblast crosstalk interaction. And you've demonstrated a couple of factors, three, but there certainly could be other factors too. And so when you start to think about, potentially targetable factors, that the question comes to mind, what other secreted factors do we not know about here? HGF has shown up in a number of secreted ligand screens. There was a number of secretome papers around 2012 and 13 where HGF, could kind of overcome therapies such as RAF inhibitors, even FGFR inhibitors. So what are your thoughts on how to approach these secreted ligands? Is one of these enough, or is ZEB1 the hub of it all, and the EMT features created? Anyway, what are your thoughts? It's an interesting idea to take these as potential targets, but I guess the caveat is, what about other secreted factors?
Yeah, first of all, it is interesting because we have preliminary data, and I'm not going to show it here because it's very preliminary, but we are investigating the role. We are going deeper into the role of ZEB1 in tumor fibroblasts because we are interested a lot in these cells. And one of the things that we have seen is that in response to TGF-Beta, that is the main profibrogenic or profibrotic cytokine that it's involved in the activation of these cells. In response to TGF-Beta, there is an activation of two EMT-inducing transcription factors. There is the activation of snail and there is the activation of ZEB1. And what we have seen so far is that the response to TGF-Beta seems to be mediated in two ways. Snail is more in charge of the transactivation or transdifferentiation of these fibroblasts. So the regulation of alpha SMA and collagens in response to TGF-Beta is more mediated by the snail. But it seems that the secretome of these fibroblasts is more regulated by ZEB1 because there are other cytokines that are regulated. We have to validate, so we don't know. But so far, for example, we cannot validate it in our tumor models right now because it's not expressed in mice, but we have done some analysis on interleukin-8, and actually in the experiments that we performed, I'm going to go back a little bit. Here, this experiment is exactly the same when you analyze the expression of interleukin-8. So there are for sure other factors that can be targeted, but what I think is that, first of all, we need to understand the role of these factors, not only in the crosstalk between tumor and and fibroblasts, because as I was telling before, for sure, IL-6 is having other functions working with the immune component, or AGF, or for example, CTGF has been also involved in interacting with immune cells and with endothelial cells. So it's important to know the actual role of these cytokines or other potential soluble factors that can be targeted with all the other type of cells to be more effective when we treat these patients with this kind of neutralizing antibodies, and to try to get the more effect, or the best effect in reducing this. And also regarding your comment about the chemoresistance, I think you are right. I think it's important to take in account the role of these factors in preventing the chemoresistance to drugs. For example, in our previous study, we observed that IGF-2 was one of the factors mediating the chemoresistance of tumor cells to anti-EGFR therapy, to erlotinib, it's a tyrosine kinase receptor against EGFR. So these factors may be involved in the chemoresistance of these tumors to typical drugs that that the patients are receiving, and this is one of the things that we are involved now, and one of the things that I'm going to comment in the next slides.
I think there's another question from the audience too, Javier, on this slide. The question from our audience is about the focus here is those secreted factors. These are experiments with the conditioned media that helped you get at those and the added factors. So what were your thoughts on studying this crosstalk in a system or model where you have direct cell-cell contact between the tumor cells and these fibroblasts, and they're suggesting that this may be more representative of the in vivo tumor. Just get your thoughts on that question and comment.
Yeah, actually, if you give me a couple of slides, you will see what we are doing now. We are actually setting up, well, we have set up already a two-dimensional model in which we mix tumor cells and fibroblasts. We are working parallel to try to add also immune cells such as macrophages into this mix, to try to understand better, as you say, in a more physiological environment, more, let's say, more in vivo environment, even if it's in vitro, these kind of interactions, and how not only the secreted factors, but the cell-cell interactions can affect this crosstalk. And also, we have recently, in collaboration with the group of Gregory Gores and Sumera Ilias, we have talked about this model a few months ago in this series of seminars. We have recently set up the syngeneic orthotopic model with the ZEB1 cells, and this is one of the things that I want to perform. I want to perform this syngeneic model in wild type and ZEB1 cow mice to try to see how ZEB1 downregulation in the stroma may affect tumor progression in an actual immunocompetent mice, because with the arrival of the immunotherapies and with the last results from the clinical trials that will change probably the panorama of treatment of cholangiocarcinoma patients in the next years, I think it's important to take in account the immune component of the stroma. So far, we are continuing this work. This is the last part of the study, is what we are doing right now. The rest of the study is what we published last year in hepatology, and this is what we are doing now. We are continuing our work with fibroblasts, and we have generated another model of fibroblasts downregulating ZEB1 also in this other LX2 cell line. And as you can see, the downregulation of ZEB1 again produces a downregulation of alpha SMA, as you can see by PCR and by immunofluorescence, which validates our previous observation in the H-tert cell line. So the thing is that I did my PhD in the laboratory of Jose Juan Garcia Marin, and one of the expertises of this laboratory is the study of chemoresistance in different gastrointestinal tumors, and I have inherited this love for chemoresistance. So I always like to analyze in my cellular models if they are more or less resistant to the typical drugs given in each type of tumor. So what we did was to expose our different cellular models to gemcitabine and cisplatin, because they are the typical drugs given to most of cholangiocarcinoma patients right now. First of all, we treated also the tumor cell models, but in the tumoral cells, we didn't find any differences in the response to these drugs, depending on ZEB1 expression. But what we saw, interestingly, is that ZEB1 overexpressing fibroblasts, control fibroblasts, were much more resistant to these drugs, to both gemcitabine and cisplatin, than SH downregulated fibroblasts. If you look at the IC50 graph from cisplatin, you can see that it's pretty correlative with the expression of ZEB1 in these cells. So coming back to the laboratory in which I did the PhD, this laboratory came up with a classification of mechanisms of resistance according to different aspects of these mechanisms. So we have analyzed different things. We have analyzed, for example, the transport of these drugs into the fibroblasts. We have analyzed the main uptake transporter and the main efflux transporters in these cells, and there is no changes in the expression of these transporters that could explain the resistance of these cells. So next, since gemcitabine and cisplatin are pro-apoptotic drugs, we thought that maybe changes in the apoptotic balance of the cells could be involved in this resistance. And actually, we analyzed the expression of a panel of genes belonging to the BCL2 family. And we observed actually something very interesting, because the downregulation of ZEB1 lead to the downregulation of BCL2, that is a very known anti-apoptotic gene. And also the downregulation of ZEB1 increased the expression of several pro-apoptotic genes, importantly BMF, that was upregulated in both cellular models. So we thought that maybe a disbalance in the pro-apoptotic and anti-apoptotic genes could make the cells more sensitive to a pro-apoptotic stimuli of these drugs. So we performed studies treating at different time points these cells with gemcitabine and with cisplatin. And for example, if we focus our attention here in this square, we observed that treatment with gemcitabine, when we treated with gemcitabine H-tert control or SH-ZEB1 cells, we observed that in ZEB1 downregulated cells, there was more DNA damage, as accounted by the expression of gamma H2AX. There was more phosphorylation of CHEK1, that is one of the proteins involved in the recognition of this DNA damage. And there was an increased phosphorylation and expression of P53, P21, and an increased cleavage of CPAR. So there was an increased apoptosis in these cells compared to the control ones. And this happened also with cisplatin in the two cellular models. So right now, where we at is here. We think that ZEB1 expression is regulating the chemoresistance of fibroblasts to these chemotherapeutic drugs. And in relation with the question from the audience, we are right now working with these spheroids, in which we mix tumor cells and fibroblasts. And as I told you before, in the future, we want to involve also immune cells. And we are working with these spheroids to try to see if these fibroblasts that express high amounts of ZEB1 are able also to, by direct-to-direct contact or by expressing some growth factors, they are able to make the tumor cells more resistant also to these chemotherapeutic drugs. And this would be really important, because as I told you before, we have found expression of ZEB1 in fibroblasts in all the cholangiocarcinomas that we have analyzed so far. So this could be a mechanism that would be happening in all cholangiocarcinomas. And if we find which could be this mechanism of chemoresistance, we could be targeting something really important in a pretty high amount of patients. So this is what we are working right now. And also we have some other perspectives for the future, because there are not only CAFs in the stroma, there are other type of cells, and we are interested also in how tumor cells overexpressing ZEB1 can interact with other cell types from the stroma, because preliminary analysis in our xenografted models show an increase, what it seems to be an increased presence of tumor-associated macrophages in the tumors that overexpress ZEB1 in tumor cells, and also an increased expression of typical endothelial markers such as von Willebrand factor, also in these tumors overexpressing ZEB1, and a decrease in the co-injection model in which we don't regulate ZEB1 in the fibroblasts. And also it has been described in other settings that ZEB1 may interact with this type of cells in the stroma. And the other way around, I told you that other cells in the stroma can express ZEB1. We have seen expression of ZEB1 in cells that seem to be in the immune component of the tumors, and also cells that seem to be in structures resembling vessels, which means that it could be endothelial cells. And also it has been described a role for ZEB1 in this type of cells. So we expect to be working on the role of ZEB1 in cholangiocarcinoma for many years. So lastly, I would like to thank both groups here in Barcelona and in Paris. I would like to thank especially Laura Fassier, my former supervisor, because she allowed me to start this project and gave me freedom to carry on with this project. And it's very nice to keep collaborating with her with this new part of the chemo assistant. And also I would like to thank Isabel Fabregat, who gave me a space in her group here at Barcelona to be able to develop a research line in cholangiocarcinoma, and more importantly even to the Ministry of Science and Innovation, because they are funding me and also my research here at Barcelona. So we have to thank our funders. And also I would like to thank the audience. Of course, I would like to thank you, Sameek, for this nice introduction. And of course, if there is time, I would be very happy to take any other question.
Well, outstanding presentation, Javier. Really elegant work, and I wanted to compliment you on the diagrams to explain these models and manipulations that you've done. They really explained it quite well. There's two questions from the audience. Okay. One was about ZEB1 expression. I think there was a co-immunohistochemistry figure or diagram. And the question was there ZEB1 in that tumor? And is it clearly in the tumors? I don't know if you could see that question. Do you know which slide that was?
Yeah. I mean, is the co-immunohistochemistry slide? Yes. Yep. It's.
I think they're just commenting that the ZEB1 was clear in the fibroblast, the cancer-associated fibroblast, but just wasn't quite sure if they were seeing it in the tumors.
You can see here the cells expressing alpha-SMA, and here in the nucleus ZEB1. Although it's true that there are other cells in the stroma that do not express alpha-SMA and that express ZEB1, as I showed you, we think that they can be expressed it can be expressed both in endothelial cells and in the immune component. And also we have data, I didn't show it here to not complicate a lot and to not make it too long, but we have isolated calves from human cholangiocarcinoma and they express ZEB1. So the answer is yes.
The second question they have is, and again, this might be a complex question because ZEB1 seems to be sort of a master regulator of a lot of things in the stroma and fibroblast. But what's regulating ZEB1? So taking step for, prior to ZEB1, what things could be regulating ZEB1?
Yeah, that's a very, very, very interesting question. Actually, I have performed some analysis. They are not here, but I can tell you that I am interested in the potential regulation of TGF-Beta 2 and ZEB1, because when you analyze the expression of the different TGF, because I work now, I belong to the TGF-Beta and cancer group, so I'm expanding my interest to the TGF-Beta signaling pathway. And when you analyze the TGF-Beta isoforms in these cellular models, the expression of TGF-Beta 2 surprisingly correlates exactly with the expression of ZEB1. So right now, I don't know yet if TGF-Beta is inducing ZEB1, sorry, in these cells, or if the cells that upregulate ZEB1 upregulate also TGF-Beta 2, but it's surprisingly coincident, the expression of these two factors. And since TGF-Beta is one of the main activators of the EMT in tumor cells, the suspect is there.
So another question about the media that these experiments and conditions are set up for. So for the spheroid experiments, there's a question that since cancer-associated fibroblasts need a specific media to grow, this may indirectly affect the normal behavior of the cancer cells in those conditions. Have you thought about the media conditions for the spheroid experiments?
Yes, actually, so far, since we are using cell lines, stable cell lines, immortalized, the condition, I mean, the media in which you culture them is not so specific. I think this is more important when you are working with primary cultures, that they are more sensitive and they need many more factors in the media that can change. And actually, we selected for these spheroids. So far, we want to broaden the panel of cells that we use in these spheroids. But for the setting up, we have chosen two cell lines, one tumor cell line and one fibroblastic cell line, that share the same type of media, of culture media. So I don't think this can affect much the role of, the interactions into these two cells. And actually, one of the first interesting things that I saw when I started to work with these spheroids is that, as I showed you with the condition media in the experiments that I performed condition media, the condition media of fibroblasts induce the proliferation of tumor cells. And when you make spheroids of tumor cells with control fibroblasts or spheroids with SH downregulated fibroblasts for ZEB1, the control spheroids grow much more. There are more spheroids and they grow bigger than the other ones. So they validate that when you work with spheroids, this interaction and this crosstalk is still working and is still there. If there are other factors improving even this crosstalk, we don't know yet, but is one of the things that we want to analyze. One of the things I want to do is to apply for funding for this project, to be able to do some kind of big transcriptomic experiment to be able to analyze with a bigger view or a broader view what kind of factors or interaction could be involved in this crosstalk.
Well, once again, thank you for this outstanding presentation. Elegant work by you and Laura's team, and so glad you're able to incorporate this concept in your work now. Thank you to the attendees and for your questions. And on behalf of the International Cholangiocarcinoma Research Network, it's been a pleasure to host all of you, and wish you well.
Thank you very much to all of you.
Take care, everybody.